what I'm going to go through today is pharmaceutics and molecule to medicine. Because a lot of the time when we think about medicines, we think about drug discovery, we think about finding the vaccine. But what you will see now is to get a medicine to patients, it's quite a long pathway. And the area I work in is pharmaceutics, and it's working on bridging that gap after discovery, and you define your drug candidate. How do you convert that into a medicine that people can use? So I'll just go through. So I like this slide because it kind of shows you the idea of molecules, which people are familiar with, but then shows the capsule, which is a medicine, which most people, the public, are familiar with, because when they think about medicines, excuse me, they think about tablets, injections, they don't think about the molecules. So what the patient takes is a medicine, not a molecule. And a lot of work goes into converting that molecule into a medicine. And that's what I kind of want to talk through in the talk today. So pharmaceutics, it can be considered the science of dosage form design. And it's not a scientific area that people are familiar with unless they're kind of working in that area and or studying pharmacy and doing pharmaceutics exams. But pharmaceutics, it deals with the process of converting that drug substance or therapeutic agent into a medicine that can be used for patients. And then when we're designing these medicines, we need to be thinking about what does the patient needs. And at a basic, a patient needs a medicine that's safe, effective, and the other term that the regulators look for is quality. Now, what quality means, it's kind of a vague term. There's no definition, there's no numerical definition for quality, but what it means is that a medicine will be the same every time. So that if you take a tablet today of a medicine and you take it a year later, a month later, a different batch, that it will be the same. It'll act the same, it'll have the same standards. So that's what they refer to by quality. Now, they're the key things that the regulators look like. However, what's coming into vogue a lot more now are these terms, and I'm interested in these: accessible, acceptable, and affordable. So what accessible means is that patients can access it. And when we look at there's a lot of really nice examples now with the COVID that you can relate to. But you know, the Pfizer vaccine, it has to be stored, I think it's minus 70 degrees. That makes it not accessible for people in countries where they don't have that refrigeration capacity. So it's limited accessibility. Whereas when you look at the AstraZeneca vaccine or the Janssen vaccine that are coming through, they can be stored in the fridge. So they're more accessible. And if we had a vaccine that could be stored at room temperature, it would be more accessible still. The other issue here, acceptability, that really refers to pediatrics and geriatrics. So for example, children, their medicines need to be acceptable. So that means a child needs to be able to take it, because children can't take tablets or capsules because of their age and their swallowing ability. So there's a need then for the liquids, and it's important that they taste well and that they're acceptable to the patient. And the same way for geriatric patients who have problems swallowing. Again, liquids unless they're viscous, aren't very suitable for them. So making medicines acceptable to the patient population who are going to take the medicine. And the last one is affordable. And while the other factors are really kind of interest to the regulators, affordable is not so much of interest, but it's of interest to the pharmaceutical companies, because they need to consider who's going to use the medicine and can they afford to get it. And that's kind of a big issue for developing countries, where some of the medical treatments are not affordable. So it's something that's also of interest. Okay, so if I now move on here. So when I talk about converting drug substances into medicines, we need to think then, what are those drug substances? And probably around the 1960s, they were mainly small molecules that were predominantly either extracted from plants or chemically synthesized. But there's been a large growth around before 2000 and close to 2000, where we are using protein drugs to treat patients. And these protein drugs, which would be hormones, a really good example would be insulin, but a lot now of monoclonal antibodies. They're larger molecules, and we're not able to synthesize a lot of them, so they're produced by cells. So we have these large protein drugs. Also, we have viruses, and now viruses are very much in the news recently because some of the vaccines they're delivering the genetic material in a virus, but they've also of interest to deliver genetic material for gene therapy. And genetic material is of interest, we see, not just for vaccines, but for treatment of diseases. And that sometimes is packaged in a virus, but sometimes is packaged in liposomes. And the most advanced medicines that we have are delivering cells to the body. So this would be cell therapies where you may extract cells from the patient, genetically modify them, and re-infuse them into the patient to treat cancer. So the drug substances that we need to deliver can be quite varied. And the ones that I suppose I've worked with most are the small molecules, the proteins, and to some amount with the viruses and the vaccines. And I'm hoping to start working more with the genetic material. And hopefully by the time my career is finished, we'll be formulating cells as well. Okay. So when we deliver drugs to the body, as well as the drug substance, we need to think, how are we going to deliver it? And there's a range of different routes, a lot of them by injection, so intramuscular, intravenous, maybe into the upper layer of the skin, subcutaneous, or as creams and ointments, topical. And they then diffuse to the blood compartment and they get distributed around the body. But we also, probably the most popular route is oral, because that's how we ingest food, and that will go down into the stomach, then be absorbed in the small intestine, into the liver, and then into the blood compartment. And then other routes would be local treatments to eyes, ears, nose, maybe under the tongue to get blood into the circulation, systemic circulation, or rectal and vaginal. So there's a number of different routes we can deliver drugs, and there's a huge number of formats that we can deliver them in. So medicines aren't all one type. We have the most popular would be the tablets, the capsules, maybe oral solutions, which I don't have there, like your Calpols or other oral liquids. We have our eye drops, inhalers delivering drugs for treatment in the lungs, creams, injections, which can be injection and needle and syringe, but it also could be a injectable pen fill. And the other areas, patches for delivering drugs across the skin into the systemic circulation. So there's a huge number of ways that we can do it. And pharmaceutics is taking the drug substance, putting it into a medicinal form that can be delivered by a route to the patient. And I suppose the area that I've worked most in is formulation. And what formulation is basically the baking or the cooking of pharmaceutics. It's coming up with the recipe of how we're going to do that. What are the ingredients and how are we going to make it? So it's a bit like baking, where you're going to get your different ingredients, put them together, and carry out a process, and then come up with your edible food. Here we're doing the same process, but coming up with a medicine. So there's other considerations around the patient health and safety that we need to consider. So what it is it's defined as the process in which chemical substances are combined to make a final medicinal product. And I've put down here some of the types of medicines I'm most interested in, or dosage forms. Microneedles, which I'll talk a bit in the talk. Tablets. I'm very interested in tablets, and you'll see when I talk about my career why that is. And lyophilized cakes, freeze-dried cakes, which are reconstituted prior to administer to a patient. So what we have is the drug, and this is mainly for protein drugs, but also it will be used for viruses as well. They're not stable long-term in liquid, in aqueous solutions, so they're generally dried. And when you're drying them, if with a protein, if you heat it up, it'll unfold and it'll change structure. So what lyophilization does is it allows you to dry something or at temperatures below freezing. So it's a sublimation. It's a specific process, but it allows you to stabilize these proteins. And I'm interested in this for protein drugs. Okay, so that's kind of the background to the subject. And I thought it was important to go through that before I start to kind of go through my career. So I did my leaving cert. I went to secondary school, did my leaving cert, and then I was debating between studying engineering, or so I was going to do science engineering or pharmacy. And my dad said pharmacy is a nice job for a girl. So that's why I ended up doing pharmacy, and I ended up going to Aberdeen. Now I don't think he envisaged that I'd go on and work in science. I think he envisaged that I'd work in a community pharmacy, and it was back in the day, it was quite I would say a stereotypical approach to your children's careers. But I don't regret studying pharmacy. But when I finished pharmacy, I worked in shops for a while, and I worked in a hospital, and I really liked the science side of pharmacy. And when I was doing the course, what the subject I liked most is the subject I work in, and I really liked understanding how things are made, how things happen, how they come together. I liked machines and equipment and understanding how they work. So that was the area I went on to do my PhD in Trinity. And when I finished my PhD, I worked then in a pharmaceutical company that was actually also based in Trinity and had a site in Athlone, which is Elan at the time. And I worked as a formulation scientist. And I really enjoyed that role. I was working with drug molecules that were poor, that didn't absorb well, trying to make oral forms of insulin and heparin. And it was really nice. I got to see a lot, and I got to travel a lot, and I really liked that job, and I liked the people I worked with. It was quite a young company and very just nice place to work and learning all the time. And I think that was really when I kind of drifted into the PhD, and at this point I thought, okay, I know now why I'm doing it, and I really like this. But as time went on, then I decided this is nice, but I want to go traveling. So I went off traveling for a year. And then when I came back, I thought I'd like to try something different. So I worked in Servier Industries in Ireland, which is a manufacturing plant. Whereas Elan developed the medicines, Servier manufactured them, and it was a tableting plant, and they made millions of tablets every year. And the parent company was in France, and Servier was in Arklow, and we worked a lot with the French people, and that was nice to see a different culture. But I really missed that R&D side, because after you've made a batch of tablets a few times, it becomes quite repetitive. So I really, my heart was in the R&D. And then a friend of mine said they're opening up a School of Pharmacy in Cork, and they're looking for people. Would you think of applying? And I applied, and I think there wasn't very many people with pharmacy degrees applying for jobs, because I got a job, even though I hadn't any kind of a research record at that stage. I'd worked in research, but in the company I couldn't publish. And I've been in Cork since. And I was involved in setting up the school, and I teach on the pharmacy curriculum in pharmaceutics, and I established a pharmaceutical research group. And what was kind of important, I think, for my background, what I brought to that was when I'd worked in industry, I could see how academia could help industry. And while a lot of groups in academia were researching, they didn't really take on board the problems the industry had. So when you had a problem in industry in Ireland, you tended to go to somebody outside Ireland to solve it, because the research going on in the university was quite focused on maybe very abstract concepts, not very applied. So when I set up my group, I thought I really want to do something that's quite applied, that can support the industry in Ireland, because it's a really important part of the Irish economy, the pharmaceutical industry. And there's lots of opportunities there. Okay, so that's the background. So I suppose my research motivation is really to provide patients with high quality, effective medicines through thorough investigation and understanding of the constituent materials and properties and behaviors. So what I'm really interested in is how we can take these materials and we can combine them to make a product that works for the patient and that is effective. And the materials that we're talking about are those drug substances, which I had in the earlier slide, but also we have materials that are called excipients, things like fillers and binders and polymers that slow down release. So there's just the agents again. And as I said, I'm really interested in tablets, which are very conventional, but there's still a lot we can do in that area. I'm interested in drugs that are poor aqueous solubility, and I'll explain about that. Microneedles, I work on microneedles with a number of people in UCC, including Anne Moore in biochemistry, and some of the other researchers in the School of Pharmacy and the lyophilized proteins. But I'm also interested in this acceptability of oral dosage forms for pediatric and geriatric patients. And in my research, formulation is my area, but I'm really interested, as I said, on the materials and their chemical and physical properties. But that's not enough. That's kind of the traditional way that pharmaceutical scientists would look at formulation. But what we need more now is I'm very interested in modeling, applying mathematics and statistics to understand interactions between the materials and the process. And also new tools, novel tools for rapid process and material characterization. There's a lot of advances in science in the area of computer science that allows us to process more and more data, and kind of brings data analysis to people who aren't experts in data analysis. But also there's a lot of advances in the area of spectroscopy and photonics and different lasers, and that can all be applied to study some of our materials that we're working with. And the last thing that I kind of focus on as well is to build on data for commercialized formulations. And I think I'll explain that a bit as we go through. So when you're trying to develop a medicine, you start off looking at the compound, which is pre-formulation. Then you're going to do your preclinical trials to look at the toxicology and the pharmacokinetics of the compound. And then we go on and try and make up a medicine and do formulation science, and then design that medicine, a process to manufacture it, and then you'll do your clinical trials. So before you can ever test anything in a population, you need to have a product, and you need to be able to produce it reproducibly. So it has to be manufactured to a high standard. And once then it's approved, you need to continue manufacturing it, industrial manufacturing science. And the areas that I work in then would be the pre-formulation, the formulation science, the process design, and the industrial manufacturing. The areas I don't work so much in is in the preclinical testing in the animals, and I'm not involved. I have been involved in the past, but not involved in the clinical trials. But other people in the School of Pharmacy and in UCC would work in those areas. Yeah. So pre-formulation, what we're doing is we're taking that drug substance and we're trying to understand its chemical properties, its physical properties, its biopharmaceutical properties, how it'll work in the body and the patient, and taking all those into account, deciding what dosage form we're going to make. And to give two examples, if, for example, the drug has a low melting point, it's going to be difficult to make into a solid tablet, so it's not going to be practical. If the drug is broken down in the stomach by the acid in the stomach, or it's not stable, it's broken down by the enzymes, such as protein drugs are broken down by the enzymes in the gut, they're very difficult to deliver orally, so they'll be injected. So you need first to consider your drug substance and what routes it could tolerate, that it would be stable in, and that will a lot of that will influence your dosage form design. So an example would be a drug we work with is fenofibrate. It's really poorly water soluble, but it's actually it absorbs well in lipids. It's used to treat cholesterol. And why I've got Venus de Milo here is she's made a marble. She's actually more soluble than this drug. This drug is really poorly water soluble. Sorry, maybe not. This drug is around the same solubility. Itraconazole is another drug we work with, and that's less soluble than marble. So this Venus de Milo has been exposed to environmental humidity and she's still intact. Whereas these drugs, we call them either brick dust or grease balls. They're just not water soluble. So why is water solubility so important? Is that the media in our stomach, in our intestine is aqueous, and this is the villi in our intestine, and to get into the systemic circulation, the drug has to break down from a solid into a molecule and then be absorbed across the cells into the bloodstream. So, okay, sorry, I'm just going to shut the door here for sorry. Okay, so when we have these drugs, there's a range of things we can do with them to make them more soluble. And we can chemically modify them. If they're ionizable, we can make them salts or prodrugs, but there's a wide range. And the one that I'm interested in is this loading drugs onto high surface area carriers. So if a drug is poorly water soluble, we need to get some way to get it to go into solution. And what we do is we absorb it onto this silica surface. Silica is really poorly water soluble, but very hydrophilic. And what we do is it's got a huge surface area. So this is an example. It's porous. It's really high surface area. It's got per gram, it's got the area of 3 tennis courts. So it's actually hard to imagine. So what we do is adsorb the drug onto the surface. So it's there, the molecules are adsorbed. So when the water comes, it'll detach and go into solution. And when it's adsorbed on the surface, it's no longer crystalline. So this is a technique, powder X-ray diffraction. We can show the drug isn't crystalline. There's different ways of adsorbing it. And this is the amount of the drug that will go into solution over time with the bottom line, whereas the top lines here are the drug adsorbed onto the silica. And this is the top line is after it's prepared and then after it's been stored for 12 months. So it's one way to make the poorly water soluble drug go into solution faster. And this is just the pharmacokinetics. So we wanted to see did it work well. So with a colleague of mine, we dosed it to pigs, and we compared it with the product that was on the market. And what we both saw, it was equivalent to the product that's on the market. We don't have the drug by itself in that study, because the drug by itself, we wouldn't capture it at all. It just wouldn't go into solution and it wouldn't be absorbed. Okay, so I mentioned that when we're trying to come up with the recipe for a drug, or what materials should we use, where do you start? And what I think is a good approach is try the tried and tested approach. If what's been in products already, what have other people used? And here was this was a study we did where we looked at all the protein formulations that are approved by the European Union. And we looked at how many were freeze-dried, how many weren't, but also what types, how many had surfactants, preservatives, other excipients. And if they use buffers, what types of buffers did they use? What types of amino acids did they include? What types of stabilizers? So just really being familiar with what's already approved, because that already works. And the other way is trying to determine how much of everything you're going to use. It can be hard. And it's not just the right materials, but the right ratios of materials. And this was a recent study. There's a theory that's out there. It's called the percolation theory, and it had been proposed, and we want to look at it for our tablet formulations. And the idea is, if you have our excipient, microcrystalline cellulose, and you start adding drug, the drug will percolate, it'll mix. But the tablet, the matrix of the tablet's mainly microcrystalline cellulose. So if we keep adding in the drug, eventually you will have more microcrystalline cellulose than or more drug, ibuprofen in this case, than microcrystalline cellulose. So there's kind of a tipping point where it starts off with the drug dispersed in the microcrystalline cellulose, and then it becomes the microcrystalline cellulose dispersed in the drug, and that's called the percolation threshold. And you can calculate this out mathematically, and the student who was working on this, she did that. And it takes a bit of work. As a pharmacist, it was a chemical engineering student who did this work, who had better mathematical capability. And she worked out the threshold. Now, what was interesting is we then looked at, okay, could we visualize the threshold? Do we have to work it out mathematically? And we looked at this Raman microscope. So Raman is an imaging is a spectroscopy technique. And what we could do is identify from mapping the surface where the drug was, where the excipient was. So this one, first one here is all drug. And then as we increase, sorry, it's all excipient. And we've two different types of the same excipient. And then we slowly add more and more drug, increasing the amount of drug, and you can see the amount of white specks increasing. And when we get to 15%, we have quite a lot. But when we get to 20%, if you took a pen, you could nearly go all the way across, joining up all the white dots. You know, there's nearly, there's a network of connected drug. And that's where you see you have the excipient, the blue, dispersed in the white, and then 30%. So this is the cutoff point. And why this is of interest is because when the excipient is in the majority, or if those particles are all connected, the blend will behave in a certain way, or the tablet. But when the drug is forming the network, it'll change. And what we see here is, this is the flow of the powder measured by variability in weight. And we can see it's pretty similar up to 15%, but at 20%, we get a large increase. We start to get a lot of variability because the API powder doesn't flow as well. And when we look at how fast it goes into solution, it starts to slow down at 20 and 30%. And if we plot the amount of time it takes for all the drug to go into solution, we've two different types here, but at 15% above it, we start to get this jump up. So it's important to know how much of everything to include in. Okay, so the next point, that's just two examples of considerations in designing a formulation, designing your medicine. The next thing you need to think is, how will I make that? And how do I make a medicine? You're going to have to think not just now of your materials, and you might have made it in a lab, but a lab process isn't the same as a manufacturing process. So you need to think about what type of equipment, what type of process parameters, and then for some medicines, you need to think about the environment, you know, the particulates or bacteria in the environment. Does it have to be a sterile environment, or the temperature and the humidity of the environment? So really what I'm going to go through here is in these three here, material, equipment, and process parameters. So I mentioned that we worked on microneedles. So how we make our microneedles, and we do this in conjunction with Conor O'Mahony and Tyndall. They prepare through silicon, very sharp silicon microneedles. From that, we produce a mold. So that's the master. We produce a silicone mold. And this is a picture of it below. And we fill that mold with different formulations. And then we dry it, and we end up with these microneedle structures. So they're solid microneedles made of material, and they can contain vaccines or drugs. And there's a number of different ways we looked at filling these molds. Now, it's very challenging to fill these molds because they're silicone, so they're very hydrophobic. And the tip here is nanometers. So we need to get the liquid going right down to the tip and filling, because there's air in them, and we need to displace the air and fill them with the liquid. And we've looked at different ways, and one of the ways we looked at it is piezo jet, inkjetting, putting little inkjet drops in. But the inkjet is designed to dispense inks, and inks don't have the same liquid and viscosities as our formulations, which would have our vaccines and water and sugars to stabilize them. So one of the things we need to look at is not just what formulation stabilizes the vaccine, but which ones can we inkjet. And I've kind of here got a range of pictures. I'm sorry, I don't know if I can move this out here. So yeah, you can see here that there's different types of, this is the inkjet, and these are all bad examples of materials that don't jet well. And maybe if I move myself down here, here we can see this is on the right is really nice inkjetting, forming reproducible droplets, and it should be in a regular stream. I'll just, so to get a material to inkjet, it's known that you need to have a certain Z number, and this is just the equation for that, taking into account the liquid's density, viscosity, and surface tension. But then to actually piezo jet, you also need a certain viscosity. So with that in mind, we looked at different combinations of trehalose and PVA, and we tried to identify which composition would give us the right Z number and the right viscosity that would allow us to jet. But as well as being able to jet, we needed to think about let's move this out of the way again, sorry. We needed to think about the stability of our vaccine, and would the jetting process affect the integrity of our vaccine? And we can see at high voltages it does. So we needed to do an assay for that. So these are the kind of considerations we need to think when we're designing our process. And here then are our microneedles, our lovely microneedles that we made. We made them red and white just for cork. No, not just for cork, but just to be able to show that we could dispense different layers of materials within our microneedles. So that's just an example of, you're not just thinking about what stabilizes the active, but you're thinking about how the material will work through the process. And also you're thinking about how the process will affect the material. So all the time looking at the materials and how their interaction with the process parameters. Once you've had that formulation designed, and you can show you can manufacture it, what's called good manufacturing practice, which is to GMP, which will allow you to test it in clinical trials, and it'll get approved for clinical trials, and you'll test it in different groups before it can be administered to and marketed as a product. So once that happens, formulation doesn't stop, because now you're going to manufacture it at a large scale. And during development, you'll have seen a certain amount of variability that can cause effects. But when you're manufacturing it year after year, you'll see new variability. You'll have more batches of raw materials. They'll be variable to a certain extent. You'll manufacture it through different seasons and different plants. And this variability can affect the quality. So you need to understand the process. So understanding the impact of variability in process on the product. So what you'll see often in manufacturing, batches may fail, and they'll have done everything the same way. But the reason they fail is something has to have changed. If you do everything the same way all the time, it should work. So, but if for something to change, the output to change, the input must change. So it may be some slight change in a raw material or in the process. And that's something that I'm really interested in, because during manufacturing, we're making more and more batches, and we should be able to capture that information to improve our process. And that's not something that's widely done in pharmaceuticals, but the regulators, the FDA and the HPRA, want people to do it, because you can learn a lot from your manufacturing batches. So this one example I wanted to show you was to do with a tablet formulation. And I don't know if this, we'll see now if I do have time. Yeah, so if we have poor flow, the weight of the tablet will be variable. And if the weights of the tablets are variable, then the amount of drug in every tablet will be variable, because if our blend that's being fed in is uniform, the amount of blend will influence the amount of drug in the tablet. So it's an important thing to control. And for those who aren't familiar with tableting, I just wanted to see, I had a little video clip there. It doesn't seem to be playing, but for tableting, if you can see here, there's a little white speck here. That is, the drug is filled in at this point. It's spread through a feed frame into the holes as the punches move down. And that determines the amount of drug you have in your tablet. And then as it moves around, the punches squash that into a tablet, and then at the end they push it out. But when we're manufacturing this, there's different tablet presses. So what we wanted to understand is what happens when we move the same formulation to different tablet presses. And this was a project with a company we did, Alchemers, And they wanted to make placebo formulations that flowed well, had medium flow and bad flow, and they wanted to see how they would perform on all their tablet presses. And they wanted to create what's called this design space by using a statistical surface response model. Now, and this, it's hard to see here, but this is the press speed, and this is the feeder speed, and each box here is a different tablet press. And the area in white is where the variability in weight is less than 1% RSD. So you can see here on this press, there's at these speeds and feeder speeds, we can get good reproducible tablet weights, and the same for, and here we have a white space here and a very good space on this model press. And then if we change the formulation and it doesn't flow as well, what we see is the space becomes a bit smaller. It actually is considerably reduced here. And then if we have a very bad flowing material, we can't actually get that weight control on this press, and it's really reduced here. And they wanted to be able to see this and develop this model so they could understand different formulations on different presses. And why they wanted to do this is when they get now a new formulation, they measure its flow and they say, well, okay, if we put it, they say the flow is passable flow. Well, if we want maximum speed, so every rotation, the press will make a number of tablets. So if we want maximum speed, we should run it on the feta at this feeder speed and this press speed. And, you know, and they would, for this passable flow formulation, the best press to get maximum speed would be this one. So after they developed the models, they wanted to check if the models we developed were right. And they brought two other formulations that they hadn't checked, and they made them on the press at different tablet speeds, and they predicted what the weight variability would be, and then they measured it, and the model in a lot of cases overestimated except in one case, overestimated the weight variability. So what it allows them to do is to, instead of just trial and error, to say, I'm putting this on this product on this press because it'll give me a good output, and it maximizes productivity while ensuring quality. So what I've tried to do, and I go through this and try and give examples. There are only some of the examples of how I've applied these 4 areas in my research and how they inform our formulation design. And how formulation design doesn't stop at designing the formulation. You really need to think about how is it going to be manufactured, because you don't have a medicine till it can be commercially manufactured. And of course, there's a lot of acknowledgements. Funding is a huge area, and most of my funding will come from the SFI and Enterprise Ireland, and I'm involved in the SSPC Research Centre, which is a combination of a number of universities working in this area. Different aspects. We have chemists, we have modelers, we have engineers, pharmacology, a range of different disciplines. And the work I've presented, these are just some of the PhD students. I've been very lucky to have a really good team of PhD students who've generated a lot of work. So Robert and Carl would have worked on the silica. Valeria worked on the lyophilized formulations. Anna Louisa would have worked on a lot of the modeling and the percolation threshold. Evan on the microneedles, and Raghu, he would have worked on the tableting processes, and he worked on that project with Alchemers and now is working with Alchemers. And also Walid Fasil, who was a researcher within the group who would have supported the work and is working in the area of microneedles now on his own projects. I work a lot with industry, Sanofi and Alchemers, 2 companies in Ireland, the scientists Sean, Tim, Raghu and Stephen, working with them. And also I've worked with a number of academics. The ones I've listed here are the ones that I work with on these projects. So a number of lecturers in the School of Pharmacy, and also Anne Moore in the microneedles in biochemistry, and with the microneedles in Conor O'Mahony. So it's a bit of a whirlwind tour of my research. I wasn't sure how to pitch it, but I hope I've given you a kind of an overview of what the subject area is. And if you've any questions now, this is the School of Pharmacy. This is Joey O'Shea, who's a lecturer now in the School of Pharmacy, and some past postgrad and undergraduate students. So I'm happy to take any questions that you have, and I hope you found it in some way interesting.
Thank you very much, Abina, for that really interesting insight into pharmaceutics and how a drug is developed into an effective product at the end, and all the important factors that need to be considered as well during the process. So thank you very much. So guys, this is just like a reminder, you know, we're starting the Q&A session now. So if you have any questions, feel free to send them into the chat and I can call them out, or else unmute your mics and you can ask them yourselves as well. There's no problem. So just to start out, Abina, where would you say that your passion for pharmacy and pharmaceutics came from? Like, I know you said at the start, you know, you went with pharmacy because your dad kind of said it was the women's kind of, I guess, job to go into. But, you know, at the end of the day, it was still one of your two choices that you had thought of yourself. So what do you think kind of got you into that?
I actually think I didn't have a passion for pharmacy, to be honest, at the start. I think I should have been an engineer. I'm convinced I should have been an engineer. And I think that's why I do the research. Sorry, I'm going to try and, yeah, that's why I think I do the research I do. But if you're going to it's really interesting to engineer medicines. So that's really the area I'm interested in pharmacy. Like, I have family members who work in pharmacy, and I can see that they're really good at other aspects of pharmacy, like counseling patients, and very good clinically. But I suppose my interest is really, I think I had a science set when I was younger, had a chemistry set, and that kind of kicked me off. But I've always loved, and I was thinking of this, like even when I was younger, I loved physics. I loved wiring things. I just thought it was fascinating that you could wire things and lights would go on. And I think I've nearly electrocuted myself when I was 5, or not 5, obviously, around 15, because I thought I'd wire a lamp for myself. So I think I think that's just the way my brain's wired itself. I like understanding how things work. And I think that's my passion, rather than necessarily pharmacy. But when you think about making things, why not make things that make people feel better, that make people healthy and good? So it's a good area to work with.
Definitely. I'd say there's a lot of people here as well that would, you know, be considering maybe going into industry straight after their degrees, or else consider doing like a master's or a PhD. So what helped you decide to do a PhD after college as opposed to going straight into industry? And do you think there's any advantages or disadvantages to doing that?
Yeah, I suppose when I finished with my pharmacy degree, we had placements at the end, and there was 2 industry placements for the whole year, and I wasn't top of my class, so I didn't get those. And I worked in community pharmacy, and I actually felt I couldn't get into industry without having experience. And so I would advise everyone to try and get experience. And I don't know if that was necessarily right. And I suppose for me, I saw it as it's either work in community pharmacy or go back and do a PhD. And I'd met somebody who did a PhD, and I thought, oh, that sounds interesting. But now, as years on, and I understand it a bit more, I think it's really important to get into industry. It's really useful to have some industry experience. And thankfully, a lot of the degree programs in UCC offer that placement. And even if you don't, I think you can get industry experience. Then it's a question of, do you do a master's or do you go in? I think if you can get into industry first, you're better off to go into industry and then come back and do a master's. Because a master's, okay, unless you want to change direction, a master's, I think the industry, eventually you're going to have to apply to get a job and get into industry. So if you can get in straight away, and then you could consider doing a master's. A lot of masters are now available part-time, and you might know more about what area of industry, because you can look at pharmaceutical industry, but there's probably, I don't know how, 100 different roles in there. So you may go in and you might start off in the lab, but while you're working in the lab, You might see somebody working in validation, you might say, I'd like to go into that. And then you might figure out, okay, what do I need to go into that area? Or you might see somebody who's working in business development with your science degree, and you might say, what do I need for that? So I think getting that industry experience either post your degree or during your degree, it's really important to figure out what masters you're going to do and why are you doing it. Because I think a lot of people may just drift into different masters because that's what most of the people on their course do. And then, you know, you're not going to do a huge number of masters in your life. So you might then at a later stage say, oh, I probably should have done my masters in this area. So I think working in industry gives you an idea about what kind of masters you want to do and what areas appeal to you. And then the other question about the PhD, do you need a PhD? No, you don't need a PhD to go into industry. Pharmacy students probably find it harder than chemistry or general sciences or other, you know, biochemistry or maybe chemistry students to get into industry, because there's not as many of them and people don't see how they fit in. But even they can go straight into industry. And in areas like quality control, you definitely don't need a PhD. The only really area a PhD benefits you is in, I'd say, technical services or in R&D. And if you really want to develop in R&D, when you move up the ranks, and I have colleagues, I have one friend who did his PhD but never wrote it up, and it has kind of hampered his career because he's working in R&D, he's managing PhD scientists and he doesn't have one. And I have another friend who didn't do it and is in a senior management position and is going back now to do a PhD. So you definitely don't need to do a PhD. So I suppose in short, my advice would be go to industry, have a look at the different types of roles there are, and then talk to people in those roles and see what do you need for that. Yeah, that's, I think, yeah. Don't just do it because you think that's what you need. Find a bit more about where you want to go in your career first.
That's some really good advice. Thank you. And I guess, you know, for anyone who hasn't done pharmacy, like, could someone from, like, say, a biology or chemistry background go on and do a PhD in pharmaceutics, like, or would you think they'd need more training in between or something?
No, in our lab, we have chemists, we have biochemists, we actually have chemical engineers. We're at the moment hopefully recruiting a math science person. So no, pharmaceutics is an applied science. It's a bit multidisciplinary. So if you come in with a strong chemistry background, or, you know, I think my internet connection is saying unstable, but hopefully it's okay. If you come in with a biological background, you know, you may be working more in the cell-based delivery, like people working on the siRNA delivery or the nucleotide delivery, the gene delivery, are generally coming from a biological background. So you definitely don't need a pharmacy degree to work in pharmaceutics. It depends very much on the projects. A lot of those projects I showed some of them are closer to engineering. The silica project, there's a lot of chemistry in that project, and the microneedles it's a bit of everything. I think you just have to be it's same with any PhD you'd learn, or any discipline you go into, you'll see that it's not very narrow. You'll be learning lots of different skills, and the purpose of doing a PhD is really it develops your problem solving ability a lot. Yeah.
That's really good to know, like, that you can go into it if you want, because it's a very interesting area, definitely. What does a day in the life as a professor look like?
Well, since I've only been a professor since November,
or like
professor, is it COVID time or not COVID time?
Let's look at non-COVID time.
Non-COVID time. It's much university life is funny especially when you're working in it. The difference between the day in the life of a lecturer or a senior lecturer professor is very similar. The only difference I would say as a professor is you get some more jobs. You have to sit on more interview panels, you know, or you get more admin if you're a professor. But it is, I suppose, the day of my life is I think I spend, don't tell my, I'm going to say this and it's going to be recorded, but I think I spend 25% of my time teaching. And the reason I say that is when I started off, I would have spent probably 75% of my time teaching, but now I have the lectures written and I'm only updating them and I'm more confident in teaching. So I would spend a certain amount of time teaching, answering emails, in COVID time, a lot more emails. And then a lot of the time it's supervising the projects, talking to people about new projects, writing papers, you know, from doing the work to getting the papers out is a lot of work, applying for grants. And then just running, the way the university runs is it's a governance structure. So organizing things to do with the School of Pharmacy or the College of Medicine and Health. And I'm also really keen, as you know, Jennifer, on gender equality and putting in Athena Swan applications for the school, and just making sure the university is a nice place for staff and students. So somebody asked me recently in admin in the university about being in the academic side. I think it is a bit of a vocation. You do have to like it, and that's, I suppose, what the day in the life is. And then I come home and I make the dinner, and I do the violin practice, and I do the tin whistle practice, and I do all the normal, sweep the floor and clean the house. So that's the day in the life of a professor. And I suppose in non-COVID times, it was nice. You could do a bit of travel and go to conferences. And now that's all done on Zoom. It's easier, but not the same.
Yeah, you're definitely very busy, but it's nice to have all those different roles within the one job. It's nice to have that kind of variety, you know?
Yeah.
You know, looking back on your presentation and kind of looking at the manufacturing process, we had this question was sent in advance, and it was, would you be more in favor of traditional pharmaceutical processing where a drug is synthesized using chemical reactions, or do you believe these will be replaced by biopharmaceutical processes, which is a more natural and biological process?
I would be in favor of the synthetic process. And I think the only reason we have the biological process is because the proteins are we can't synthesize them. They're just too complex to synthesize. I think with time, if we look back at it in 20 years' time, the biological process, we'll have figured out how to do it. I was talking to somebody today and they were asking me something about anti-snake venom. It's how they generate it is they infect horses with the venom, and the horses produce the antibodies and they collect the antibodies from the horse. And obviously that's not an ideal way to make any medicine. And we were just discussing that. And insulin previously was extracted from porcine, from pigs, and there's bovine insulin as well. And now that's made by cells. And I think what will happen is we actually will figure out more and more of the mechanisms of the cell, and eventually we'll be able to synthesize these outside the cells. So I think that's very futuristic, but the reason why I prefer the synthetic approach would be because it's more controllable. It's more reproducible, more controllable. And as I started off about safety and efficacy, quality is related to those. And in a biological process, there's more variability. And as a result, you can get more variability in your product. Now, the products are tested and they're safe, but I think it would be better if they were all synthesized. Because I think if we got to the point of synthesizing them, we would understand the chemical structure, the physical structure of these much better, and we would understand much more how they interact with the body. There's always a risk with a biological process that something will be carried over from the process into the product, and that's where all the quality steps are to make sure that doesn't happen. So much more in favor of the synthetic drugs, but the reality is we can't do it, and the protein drugs are excellent drugs, and they're much better, they work much better in the body. So we have our biological processes as they are at the moment. So while the other one would be ideal, we're not able to do it, but we will be able to do it. And you see that the siRNA now is, sorry, the messenger RNA, that's all chemically synthesized, that's in the COVID vaccine. And, you know, we're developing our manufacturing capability all the time. Yeah, so we'll understand more and more what the cell does, and that will allow us to reproduce that outside of the cell. So that'll be good, I think.
That's a very interesting perspective on it, actually. Yeah. You know, again, like looking, you know, at the whole process of getting a molecule all the way to, say, a medication on the market. You know, it's, as you said, like it's quite a very long process. But what would you say would be like the biggest challenge that, you know, researchers and the manufacturing companies will encounter along that time frame?
The biggest challenge is the failures. And the main reason that drugs fail is they're not effective and they're not safe. And, you know, to get to the clinical trial, you know, you can have, I don't know what the stats are now. It was 40 million a few years ago, the average cost of getting a product to market based on the number, taking into account all the failures. So to get a product to market, you have to go, well, to get a product to the clinical trial phase where a lot would fail, you have to go through all the formulation development, the manufacturing, the process development. And some medicines just don't work. And they have to be better than what's out there on the market, or they have to be as good. You know, in a lot of cases, they actually have to have some unique, you know, you can't just keep bringing products to the market that are the same as what's on the market. You have generics, but for new medicines, new chemical entities, they have to have some benefit, and that all the work has to go in up to that point, and it's only at that point you'll find out, does your medicine succeed? So it's a big gamble, you know. Not like you will be confident, but you just don't know till you put it into the patient if it's going to work as well as you want, and that's where they fail a lot of the time. And then when they get, that's probably why manufacturing is kind of neglected a bit, because that's where all the focus is getting past that clinical phase, getting it approved, and then they say, oh, now often manufacture it. And we see now with the shortages, there's a lot of drug shortages in the world, that there's problems carried through to manufacturing that aren't resolved. But while I'd be interested in that, I think the biggest challenge is getting is the costs building up all to that first to the clinical trials. And we're kind of spoiled with the COVID vaccines, because every single one that seems to be coming out seems to be efficacious. That's not typical, and I think people were very surprised with the vaccines for the COVID that they're all so good. It just shows that they're building on the science that came before. But for a new drug to treat a new disease, you really don't know, and there's a lot of failures at that point. And that's why it's very difficult for an academic group in a university to bring a product all the way through, because they need investment and the costs of that. So I think that's the biggest one. Yeah.
Definitely a very challenging process anyway, but again, like you said, it's all about the trial and error, and I guess that's the very, you know, the science heart of it, you know. And you just
well, informed trial and error. It's a very well educated gamble.
Exactly.
Very well,
exactly. And just as a concluding question, if there's no other questions that come in, what advice would you give to the audience tonight with regards to moving forward with all their STEM careers?
I think as you go along, you discover what you really like, what interests you, and I think you have to be brave and just give it a go and talk to people in the field, and don't talk to more than one person. Don't take the first advice if somebody says, oh, you have to have this and you have to have that and you won't ever do this. Ask a number of people, because everybody comes with their own biases. So ask a number of people, and I think you do have to follow your gut. follow your instinct. I suppose when I started in UCC, I was interested in tablet manufacturing, and I think 2 or 3 people told me, don't waste your time doing that. You'll never get funding, you'll never get a publication. And I did. I kept going because I thought that's what I know a lot about, and I think there's something there that needs to be done, and it paid off. But if I'd listened to those people, I probably would have gone to making nanoparticles. And I know while they're really interesting, half the world's making nanoparticles. Sorry, Jennifer, but different types of nanoparticles, half the world's making them. And it would have been a lot of competition for me. So that's one bit of advice. The other bit of advice I got that I really kind of stuck with me is years ago, someone said to me, always do technical. I was thinking of moving into business development, and they said, no, you should stick with technical, because in a technical role, you always develop your knowledge and know-how, and you're learning all the time. And that's hard to replace. While in a business role, you can always be replaced by somebody who's just better at business. And, you know, there's still skill, but it's hard to replace expertise, and you build on it year on year. So that was a bit of advice I got, and I think it was good advice, and I've always tried to build on what I did before. So you can change direction, but try and build, don't just build, what would you say, pivot, rather than just totally throw something out and move a completely different direction. Like, for example, if you were doing science and you really liked English and you thought, well, why not do science journalism? You know, try, or if you like I did pharmacy and I really wanted to be an engineer, and now I'm a pharmacist and effectively working in an engineering field. So pivot. So I suppose that's my bit of advice for what it's worth.
Yeah, that's really good advice, definitely. And I guess it's just really reassuring for everyone here that, you know, just whatever you're doing now in college doesn't mean that that's what you'll be stuck with for the rest of your life. You can really just like move around. So it's really good to know that.
I think more so nowadays. And like, I teach in UCC, but I also teach a lot of adults, teach undergraduates. But I'd say the average age of the student I teach in the postgraduate is, I'd say it's 40, and they're all people changing careers or upskilling or moving in different directions. But you know, if you're in science, you can. I think that's why I say work, because when you start working, you see what's available out there. It's hard when you're doing a course in college to know what your job is going to be like. It's like when you're doing your leaving cert, it's hard to know what college is going to be like. Will you like this course or that course? But you still make a guess. And there's a very nice thing in Alice in Wonderland where someone's trying to decide what to do, and the Cheshire Cat's up the tree, and the Cheshire Cat says, well, if you don't know, it doesn't matter which way you go, because once you go somewhere, you'll figure out if that's the right place, and if it's not, then you'll go somewhere else. So it's never the end, it's never your final destination. And who knows if I'll be a professor in UCC in 5 years' time, maybe I'll pivot too. Yeah, but I'm happy enough at the moment.
That's really good advice. Thanks so much, Abina. I guess I'll wrap things up there. So on behalf of UCC Science Society and everyone here tonight, I would like to thank you sincerely very much, Abina, for a fantastic and insightful evening.